this video I show you how to predict a protein monomer structure with bound ligand and cofactor using Protenix Server. The Protenix Server is an alternative to the AlphaFold server, which allows you to predict monomers and multimers with bound ligands, cofactors. Besides proteins, it allows you to predict DNA, RNA structure, and also add various covalent modifications to those structures. Just like any AI-based method for structure prediction, it works best when structures similar to your structure were contained in a dataset on which such model was trained. In case of proteins, it worked best for conservative proteins, and with less accuracy it can also predict disordered regions. Unlike AlphaFold server, you can use Protenix output in docking studies, but you still can't use it for training other deep learning models and for developing competitive tools. For more information, please check the service agreement of Protenix server. The web interface of Protenix server mimics closely AlphaFold server interface. You can specify the name of the job, decide which version of Protenix model should you use for predictions. You can decide whether to perform multiple sequence alignment, use a template for modeling. You can also upload the actual structure from which the required information will be taken for modeling. Here I paste protein sequence directly from pasta file downloaded from Unip database. Here Protenix server refuses to take this sequence as an input because it contains end-of-line characters. So you must first remove those characters either by using a text editor like Notepad++ or by writing your own scripts which will automatically batch process multiple such sequences. In my previous video on AlphaFold server I used one such script, so here I will do it manually. Both AlphaFold server and Protenix server expect to receive protein amino acid sequence in one letter code as one line. Here I can also add post-translational modification to my protein, which I don't want to do in this case. You can add more structures to a complex by clicking the Add New Sequence button. It can be both amino acid sequence, or it can be DNA, RNA sequences, or you can also add chemical structure of a ligand as SMILES code, or provide its ID from PDB. Or you can add ion, for instance, as a cofactor if you know that it is required. In this particular complex I have added HTF as a known ligand of my protein and biologically relevant cofactor magnesium ion to stabilize the phosphate groups of guanosine 3-phosphate. I also use default parameters of model. Once you are ready, you click Submit button to send your job to the Protenix server. As you can see, all the data that you have just defined is being converted into the JSON file and provided to the Protenix server so it can start modeling. And this input configuration will also be preserved in your output. You can find all the predictions that you have submitted from this particular email address, both already completed and currently running, by clicking on the prediction history. Here you can also track down the progress of your prediction and check out different information about it. Depending on the workload of the server and the complexity of your job, it may take the Protenix server a while to process your data. But from my experience, the waiting time is usually within 10 or 15 minutes from the moment you submitted your job. You can periodically hard refresh the web page to check out the progress on your one or several jobs that you have submitted. Once the job is finished, you can download your results by clicking the respective button, and it will be downloaded as an archive. Inside the root folder of your archive, you can find the multiple sequence alignment folder, a folder with predicted structures. You can also find license information and the input JSON file which was used to perform the prediction. The output structures are stored in CIF format, and each of them also has the dedicated JSON file with confidence information. Finally, we can check one of the output structures. I can clearly see that the globular conserved part of the protein was modeled correctly, and the ligand and the magnesium ion, which is a cofactor, are also positioned correctly in the nucleotide binding site. As for the disordered region, it is obviously of low confidence, as the training dataset likely contain almost no disordered parts. Those are very hard to determine experimentally, but in this case it is actually of no difference to me, because I am not modeling anything related to the disordered regions. So that is basically it. Thank you for your time and happy predictions.